All right. Okay. So welcome back to Neuroccino, everyone. As usual, the deal is that today I will present a paper, and sorry, I'm not as good as as Steffi for this kind of introduction, but so the rule is someone presents a paper, and then that we got interested in, and then we ask after presentation, we ask a question, we have some discussion. We are also live streaming on YouTube, so if you have question there, you can just ask them, and we'll relay them online on the Zoom meeting. All right, so without further ado, let me share the link to the paper that I have for today. This one won't work. So at least here is the DOI, because the link is not working well for me. Sorry about that. Here's the DOI. And here is the article. All right. So there's so many things on this screen. Sorry, it's a bit annoying. I'm just trying to, all right, getting full screen. All right, so this is a research paper published in Science very recently. I think it was 2 weeks ago or something like that, which explores a new method for in vivo direct imaging of neural activity at high temporal spatial resolution. And so what's the big deal here? Well, as you may know, if you are doing fMRI, usually when we scan when we scan someone or an animal, it's the temporal resolution that we have. Well, we have a quite good spatial resolution depending on the machine, with voxels that can be about half a millimeter or even sometimes smaller in on the edge side. But when we look at temporal resolution, we rarely go under half a second for each time point, between each time point, which, well, it's fine when you're trying to see broad scale kind of activity in the brain. But when you're trying to get to the neuronal activity, well, that's far from fast enough. We need to get to the millisecond. And this is actually what this paper achieved just with MRI. So here they're using a 9.4 Tesla MRI scanner on mice. So if we do this on human, we would not need to have this kind of super high Tesla power MRI scanner, but still, it's quite high power scanner. I think maybe we could do that with a 7 Tesla, the equivalent with a 7 Tesla in human, but this is just speculation for now. We'll see later. So they scan mice, and they do it so there is a few things that they did to try to reduce the time between each scans. Is first they do a 2D scan. Basically, they just do a scan through a slice of the brain instead of the whole volume, which helps obviously. But also they designed the whole method so that it's actually it's a clever scan. It's clever scanning procedure. It's instead of scanning the whole brain or the whole slice of the brain at one point during the scan. I mean, to have the whole thing, they actually scan only. It's a bit complicated to explain it like this. Sorry. They have a repeated stimulus. Here it will on the mice, it's an electrical stimulus on the whiskers, and the repeated stimulus is done every every 500, I think, milliseconds. It was written here. Where was it? Oh yeah, it was, sorry, 200 millisecond interval, so 5 times a second basically, and their TR, so the time between each acquisition that they do is 5 milliseconds. So 5 milliseconds, super fast. But actually, what they do is not to scan the whole brain for each 5 milliseconds. They scan only the first line, like one line of the K space. The K space is a space that is used after to reconstruct the 3D image or the 2D image here of the MRI. So it's not like real scan, like real thing that you can see usually. It's data that it will be transformed, but doesn't really matter here. What matters is like they do the first line of the first time point during the first 5 milliseconds. Then during the second 5 milliseconds, they do the second line, etc., until the 40th time point in their time series, because the time series is defined, like it will be 40 frames here. And so this was done with one stimulation. Then there is a second stimulation, and they scan the second line for each time point. So they have to have a very repetitive stimulation, and with that they assume will give the same or approximately the same result in the brain that they will scan. Because as you see, you do for example, for the first time point, in the end, the first line will be from the first stimulation, and the last line will be from the last stimulation when they reconstruct when they create their K space here. But in the end, that means also that they manage to have time points in the time series in the scan that are separated by only 5 milliseconds. So, but how good is that? How do they compare with what exists? So here you can see the brain, like the brain slice that they are investigating, and they chose a place in the barrel cortex, where the whiskers are supposed to activate this region. And so here you have just a plot of the signal received in this area with, so they call their method Diana, so Diana fMRI. And so, well, there are the two controls that are staying around 0, and you have a nice spike here after the stimulation, which is at 0. Here a few more ways to show the graph. So you have a clear activation, but then they asked, okay, so how does it compare to actual physical data? And so, sorry. So they did some in vivo recording with more classical methods. So one was with fluorescence, and one was direct unit, single unit recording with electrodes. And what they get is basically, so with the LFP, so the fluorescence, you can see that you have after each stimulation, you have a decrease in fluorescence, I guess, or maybe not fluorescence. I'm not super. I didn't really look into that, but you have a decrease in the signal. And when you check the peak latency, it's quite different. It's quite high compared to there's like 40 milliseconds after stimulation for the peak latency, compared to if you do the single unit recording, where you have only about 25 milliseconds between the stimulation and the spike recording in the cell in the cortex. And then you check, but what about Diana? Well, Diana has exactly the same or about the same latency as single unit recording. So we are really recording the electrical impulse, like from the neuron spiking here, which is, to me, it's quite crazy that we get an equivalent result between single cell unit recording and the just MRI scans. So then another experiment they did was to say, okay, hey, that's cool, but can we check also the thalamic response with everything? So they took another slice with the thalamus and still the barrel cortex and checked what they got. And actually, here you can see in green is the spiking they managed to detect in the thalamus, and in pink or purple is the spiking with diastole that they had in the barrel cortex. And you can see that nice spiking in both, and with first the thalamus spiking about 10 milliseconds after the stimulus, and then again about 25 milliseconds after there is spiking in the cortex. They checked, and yeah, okay, so, and what about if we do it again with a single unit recording? And then you have the result here again, and you have the same kind of things, meaning first the thalamus, 10 milliseconds, okay, we have the same thing here, then the cortex, 25 milliseconds. So it is really, really neat at this point. Then they did the last experiment, I think, where they tried to do, they did, optogenetics to do the stimulation, and while to stimulate directly the barrel cortex and to see the response in the thalamus, and it worked as well, and with a very, very short difference between like the stimulation and then the response in the thalamus. So that's basically it. They did a few other things also to check if it was related to the bold, if the thing that they were recording was related to the bold, which does not appear to be the case at all, because they did experiment with mix of air and oxygen and just air to see if it would change things, which would in bold with the bold signal, which is the classical fMRI signal. And actually they don't really see much changes in their signal. So it seems to show that the thing that they are recording, so it works. It's super fast. It's not bold. It's they have hypotheses on what it may be exactly. I won't go into the details here. But it's just hypothesis. Nobody is really truly sure yet. But it's not. To me, it doesn't really matter. What it matters is that it works, and it's crazy interesting. So next question, of course, is can we use it on human? Well, we'll have to see. Also, because it's only on. It requires a scheme that is quite specific, where you have a very repetitive stimulation. It will only be able to be applied to very specific, I think, questions, but to me, it's still so cool. You have basically a proxy of cell recording, but you don't need to implant electrodes in the brain to do that, to get this signal. You just need to have a very fast MRI, a very good MRI, and that's it. So what do you think? All right, so let's go back to this screen. And I see there is some, sorry about that. There is the magnet. All right, sorry about that. I need to go back to Zoom. All right. And close this. And so Michel has his hand raised. And I think there were questions also in the chat that I didn't check.
That's the same. That's me. I've been taking my notes in the chat.
Oh, okay. All right.
I can ask one, and then Chris will follow up, and then I have some. I'm sorry, but
yeah,
I am not sure I understand exactly what the acquisition is. What is a contrast?
I don't think they are using a contrast specifically. They are not looking at the contrast. They are looking at a difference in activity, basically in the voxels.
Right, but how do they do it with the MRI? How do they measure it?
What do you mean? It's like every voxel, well, here is a pixel because it's in 2D, but every pixel has its own time series, and you just look where there is change in the signal. And
but what is that signal? Because usually you look at bold, but here it's not bold. So what is it?
Oh, well, that's the question, right? I mean, it's one question, but does it really matter? It's
no, yes, it does matter. What is a part of the signal from the MRI that they were interested in, like to extract these values that correlate with neuronal activity? You have different kinds of sequences. What did you use exactly? That's what I don't understand.
I need you to repeat a little bit that, because sorry.
So like with your MRI, you can measure the diffusion and model it. You can measure the bold and model it. You can measure the T1, the T2, and like a bunch of different sequences that you can use. Here it's and you highlighted it's not bold. They don't know exactly what it is biologically, but I'm not sure what the sequence is. I missed that point. You probably say that I missed it.
No, I didn't really say it because I'm not super.
Bold.
Yeah, bold is just the biological explanation of a signal that is measured. But what they do exactly? They use a sequence. I don't know exactly. No, I think it's called the fast sequence. And they have it has its own TE and its own TR and its own thing, and it's it's a 2D echo gradient imaging sequence that has already been published. It's something that is, I think, quite classical in this kind of field, but is
it related to what Le Bihan was doing with the diffusion imaging?
I have no idea. I don't think so.
What molecules change?
I don't think so. I'm not super. I don't know well about what Le Bihan was doing. I've heard a bit, like briefly, but not that much. But I think for them, when they did their experiment, they say that it's related to some kind of properties of the water on membranes of the cells that changes, and that change probably the magnetic properties of the water near activated cells or something like that. I'm not
okay.
But this is just the biological explanation. Yeah, so it's just, to me, it's like the bold signal for a long time. I think for a long time we were not sure what it was exactly either, and so
I see.
It's just a signal that we have a machine. We manage to see that there is a change in the signal when we do stuff, and we are like, okay, so then we try to explain where it comes from. But most importantly, we see that it correlates really well with the experiment, and we can use it to try to explain how the brain works, which is mostly what matters. If you can prove and explain why it does it, it's super interesting. It's a really nice question. But to the problem of how the brain works, when you do a stimulation and where it goes, etc., I don't think it's a critical question.
I see. Okay.
All right. Did you have other questions? No. All right. So, Chris.
Yeah, thank you for presenting the paper. One thing I might have misunderstood, or maybe I didn't hear. How does that work? Because so you explained that they are taking as 2D slices.
Yeah.
But also in the figures, it looks like they are interested in very specific regions. So are they like to make a fast acquisition? are they taking just one part of the brain that they are interested in, or
No,
are they scanning the whole brain every time?
no. As I understood it, they're looking at one slice. They're not looking at the whole brain. They say, okay, we will look only at the slice that is this way in the brain. So it will be a 2D scan. You won't have the whole brain, just a 2D scan, but they do scan the whole slice.
Okay.
And then on this slice, they select the voxels that are in the barrel cortex and say, now what is inside here? How is it spiking? And they can plot the thing that you have here.
So, I mean, of course, on mice, one slice, even if it's a half a millimeter, half a millimeter, if the voxels are half a millimeter of resolution, you still capture quite a big proportion of the mouse brain.
For human, when I was saying half a millimeter, like earlier, I was thinking of human scanners. With the 9.4 Tesla and on mice, usually it's much smaller. We are in orders of like tens of millimeters or even smaller.
Yeah, so when you're looking at a region, usually a region is bigger than that. So can if they want to look at the activity of the whole region, would they do several acquisitions with the same stimuli and just like aggregate the slices?
Wait, I'm not sure.
Because if they take just, you said they take just one 2D slice. Yeah. But if the region that they are interested in is several slices wide.
Oh, okay. Yeah. Well, technically, because of the way they are doing it, they could very well, I think, do several slices. But they would have to do it like first one slice, then another slice, etc. But when you think about it, this is also kind of what they are doing, because they are measuring the first row of the K-space first for every time point, then the second row, etc., etc. So it's already, you already need to have the stimulus that is locked on the
repeated
the scans.
Over several,
so you could imagine to repeat this for different slices and get at the end a 3D scan. Exactly.
Yeah. I mean, it would probably be very difficult to have the whole brain doing that. But if you're interested in a bigger region than just that doesn't fit on one slice, it might be possible on several slices.
Yeah.
But that's super cool.
All right.
Thanks.
Now we have Michel back. Michel, floor is yours.
Sorry. So I checked, and that's actually, if I understand well, that's actually following like what Daniel Bion have been doing, which is looking at the water diffusion and the movement of water molecule. And the idea is that because the neuron, before a discharge, like is doing this kind of, change of movement, like the water molecule around the neuron will change the profile of diffusion, and that's how they can measure it. But the problem is I tried to do that in Paris, and you can only have like a few slices of the brain. You cannot cover the entire brain. So this K-space approach is like very smart, because you can have the full brain and you can have it faster than if you were doing, the normal acquisition that is recommended. I guess my question is, so you talk about the K-space, is form of like an embedding, a pre-embedding of the data, so you can reconstruct like the entire brain, and smart way of acquiring where you have most of the information. I was wondering whether you have to know the K-space of your own individual, or is that a generic K-space that you can apply to any brain?
But I think the K-space is not some kind of embedding. It's more like the way the signal is retrieved from MRI data is encoded both in phase and in frequency, and not in real visually accessible data that you can have on your screen. And this is like just the way MRI works. So this will be the same kind of probably, I'm not sure, but I guess it will be the same kind of k-space for any machine and any scans. And because you just need to do just probably more things that under the hood, but it just a 2D reversed Fourier decomposition, I think, that you do to go from k-space to 3D space.
I see. Okay.
So, but yeah, I'm not a physicist, an MRI physicist. If Leah was here, maybe, but she's probably sleeping right now. It's
I just have questions, you know.
Yeah.
And then I had another question regarding this significant relationship with spikes.
Yeah.
The same thing. I was writing my question, so I'm sorry I didn't hear all you said. Is that because there is no significant difference between the spike and the dianas, that they said it's the same?
So they are careful in the way they say it. They say we did not manage to find any significant difference between the two, and so they are likely equivalent.
Isn't that like a violation of frequency statistics? You know, there is this entire
but you can speak about like difference in you cannot say they are the same if they are not statistically different. It's not the same. It's true, but you just look at the data. They are really close anyway. Even if it's not exactly the same and there is a small difference, which is possible, the fact that they are really, really close is still just what we care about in the end.
Well, what's surprising is like the latency between the peak of activity. It's not like a single neuron activating. The peak of activity of a group of neurons that follow a Gaussian distribution is the same. Although I like if you have a look at the Le Bihan experiment that he's been doing, he always said that his diffusion fMRI peak of activation was always earlier than the classical Bolt signal. But that's pretty cool if that's at the same time as a neuronal activity.
It's true that it will be anyway like faster. Using that will be faster than the Bolt signal anyway, because we know that the Bolt signal takes sometimes seconds to start to really show up, and here it's a few milliseconds. So that's for sure. But honestly,
because this work from Logothetis would try to see whether this was really the activity of neurons. So he was saying that's more like the blood flow, that because also like when you have more blood flow coming, it shifts a little bit like all the vein and arteries in the brain, and it moves the water molecules. So it was like, okay, also you're actually measuring the blood flow. I don't know.
I mean, even if it's the blood flow, but
the blood flow should arrive after the neural peak.
Yeah, and the fact that it is so closely related to the spikes here, to me, it feels unlikely that you'd have a biological response with blood flow increasing and everything that fast. That seems unlikely.
That's impressive.
But then again, it's time will tell exactly what it is. I'm more interested in the application that we could have rather than the exact understanding of what it is, personally at least. But I encourage you to read the part where they tried a few things to disentangle where it comes from, because they did some stuff, and I went quickly through it, so I'm not completely sure about that, what they did, but they did try to demonstrate what it may be, and they have a few hypotheses that are confirmed by that seems to be aligned with their experiments. So yeah, we may want to have a look at the last part, which it's a quick read. Honestly, the whole article is really nice to read. It's short, it's dense, it's condensed, and it's really a nice read. But then in terms of application, for me, it would be really interesting into looking at not only like the thing that they showed, like here, the time difference between activation in the thalamus and in the cortex, which is nice, and things that you cannot really see with the Bolt signal because it's really too closely related, but more like inside of the same. One thing that would be interesting to me is like inside of a same region of the brain. If you have enough spatial resolution, can you see like activation coming somewhere, and then maybe in the same area, let's say like if you do finger tapping or something like that, does it have its own little network inside of the hand knob area, and saying, oh, but actually it's not just activating as like a big blob as we see in fMRI, but there is like going left and right, and then up and down, and then going to the thalamus, and then maybe going back, and all of these kind of things that are super fast and that we cannot really see right now. But I'd be super interested by that.
Sandrine just uploaded the supplementary material. 50 pages.
That might not be as short as the original article.
Yeah.
But thank you, Sandrine.
Just trying to figure out exactly the contrast that they use. Yeah. Oh, that's fascinating.
Yeah. All right.
Oh, Valentina has a question.
Oh, Valentina.
A really fast question, because I know we are running out of time.
Sure.
So with this spatial and temporal specificity, could we use this to explore the functional signal within the white matter?
Well, that's a good question. I have no idea, because
it was a fast one, no?
yeah. I have no idea, because the functional signal in the white matter is still quite new. Well, new. It's done by a few labs, but not broadly done in the field with bold. But here, because we're not looking at bold, I would be curious, to know if we actually may see better stuff with this kind of signal than in the white matter. I have no idea. I don't know if
whatever the signal is.
But that's a really, really good question. Yeah. All right.
To be explored. Thank you, Victor.
Well, I think we have to wrap it up because we are running out of time. So thank you, everyone, for your question and your attention. And I haven't checked every question on YouTube. Well, sorry if there was, but at least thank you for attending, and see you next time. Bye-bye.
The YouTube questions will be addressed next time.
All right. So.
Still live? Are we still